I am an RNA biologist, and I have studied messenger RNA, mRNA, since before you were born, since 1972. And
That's before I was born, Lynn.
oh no, it is not, is it?
1975.
Oh my God.
Sorry. I couldn't
You
hold that
had
back.
to say it, didn't you?
Sorry.
Oh my God. That makes me feel odd. All right. But I don't look it, do I?
You definitely don't look it.
I went to the gym yesterday. Oh my God. Anyway, so I've always known that RNA was important, and I've really loved studying the molecule. These days, we know most of our DNA, our genome, is making RNA, all kinds of different RNA that, depending upon the type, can have all kinds of functions all over the cell. So it can be an enzyme, it can be regulatory, it can be involved in really important cellular processes, including encoding the proteins that our cells make. And the way that mRNA is generated gives us the protein diversity that we need during our development. Think about our development in utero. I mean, this is true for all organisms that are mammals, let's say. And as adults, think of all the different cell types that we have. So between the importance of RNA in our existence as human beings and how RNA metabolism, when it goes awry, contributes to human disease, and now the horrible pandemic we're in that is caused by an RNA virus that can be treated with a vaccine that has a little piece of the viral messenger RNA in it, I think the stars have aligned for lots of people beyond those who are RNA biologists to realize the importance of RNA. I'm done, so you can go, Josh.
Okay. So I study how viruses kind of modulate cell biology to support their replication. And our lab really focuses on two areas of this. Primarily, we try to understand how viruses kind of interact with the cell to drive the production of the biomolecular subunits to produce more virus. So viruses are obligate parasites. They rely on the metabolic resources of the cell to produce energy and to produce more things like RNA, and they produce them in mass quantities. And so there's kind of like this, host pathogen interaction between viruses and the cells, and the viruses have to take control of those metabolic resources. And so the other aspect of the lab, besides just kind of control of metabolic resources that we study, is also cells have kind of like innate immune kind of pathways that are always trying to recognize when viruses might be present. And so there's a lot of interactions between the cell trying to recognize foreign nucleic acids, for example, RNA, or foreign DNA, or even foreign lipids, and then react to it to try to shut down that cell and signal to the immune system that the immune system should come and kind of consume that cell and process those antigens so they can identify other cells that might be infected. And so there's a lot of viral genes that are responsible for counteracting that process, and a lot of cellular genes to try to prevent it from counteracting with that process. And so we study various contexts, typically in herpes viruses, but when the herpes viruses, whether it's herpes viruses cause herpes simplex virus, but also mono is caused by, and chickenpox is caused by herpes virus. And there's been herpes viruses associated with chronic fatigue and a bunch of other kind of syndrome-like illnesses. But once the pandemic hit, we were really interested in coronaviruses, and so we got a couple of different strains in SARS-CoV-2 and some less pathogenic beta coronaviruses, and we had been doing some metabolic screening with anti kind of metabolic compounds in an anti-cancer context, because I collaborate with a lot of people doing anti-cancer, and so we quickly tried to, we successfully then converted that into an antiviral screening kind of approach. And so this isn't so much vaccine development, but really looking at compounds that you could use to potentially treat and repurpose compounds that are already been used for other purposes to treat coronavirus disease. So, and we first started out that because the screen was very large with a less pathogenic coronavirus, but then with our hits, we went into a BSL-3 facility, which is required to go against SARS-CoV-2 and work with SARS-CoV-2. And then we found about half of our hits that were active against the less pathogenic were also active against SARS-CoV-2. And so that's kind of where we are with that at the moment.
Did you isolate RNA from those cells?
We did. So RNA.
Doing RNA-seq, because, so here's the thing. This is kind of an aside for you two. So I will tell you though that one of the defense mechanisms that the cell has against viruses is the pathway that I'm most known for having discovered and worked on. It's called nonsense-mediated mRNA decay. And so, you know, Josh, in talking with Sarah Cherry, if you look at the transcriptome of cells that are infected with SARS-CoV-2, there's no evidence for the virus inhibiting NMD.
Huh.
Have you, and so she said, she wonders, she's a virologist who's a colleague of ours, and Sarah wondered if that's because they replicate like crazy. So this virus replicates at a faster and higher rate than other viruses.
Yeah.
For RNA?
I haven't looked at that. I mean, primarily what we're just using the RNA as is a readout for how quickly the virus is replicating, because it's a great correlate. But I think that's really interesting. So it's
and we show that when we expressed N protein, we actually promoted NMD, which I don't know what that means. But anyway, that's an aside, except for the NMD part.
So.
Okay.
I mean, our work isn't directly related to the vaccines, although Lynne, you know, is one of the global experts on mRNA biology. So it's like in that regard, you know, it's like her and basically, I don't know, her cohort of. What'd you say? Yeah.
So we just won the Wolf Award. You should look up the Israeli Wolf Award.
Yeah, her good friends have been like kind of pioneering, you know, the technology that went into the mRNA vaccine, and it's really, I think it's really blown away all kind of expectations about how well it could do, and it's going to revolutionize vaccine biology without question.
Although, to answer some of your questions, this vaccine development did not happen overnight.
No.
It has taken decades. And so first we had to understand about how mRNAs are synthesized, how they encode proteins. So we had to understand how normally mRNAs are made and function. And of course, they're made of nucleotides, and we had to understand what are the optimal nucleotides for the synthesis of protein. And mRNAs are generated by complicated processing steps. So we had to understand which of those steps are important. If one takes an mRNA, such as that's in the vaccine, and think it's going to be stable when injected into a human person, so it can make enough protein so that it evades the immune system that Josh was talking about. And so it also took decades to figure out how to incorporate that mRNA into nanoparticles that can be taken up by cells, that don't get gobbled up or its contents gobbled up by the macrophages, so it can get into our cells to make protein. And one of the big discoveries is the fact that if you modify mRNA with pseudouridines, so you can make your RNA in a test tube with pseudouridines instead of uridines, then the mRNA will not be recognized by our immune system and destroyed. So we want the virus to be recognized by our immune system because it's a bad thing. We don't want the mRNA in the vaccines to be recognized by our immune system because we want it to be stable to produce spike protein. But this process of packaging it in nanoparticles and making sure the mRNA can be translated efficiently, making enough mRNA for vaccines, these incubation reactions that make the mRNA that's incorporated into the nanoparticles, that then they are enormous in size. Like they're the size of a desktop, a giant desktop. They're amazing. So this all took decades of work.
So could you talk about a little bit of some of the different technology that you've helped develop with the mRNA vaccines, and maybe like how that differs with like the Johnson & Johnson vaccines as well?
Do you want to talk about that, Josh?
Sure. So, I mean, I can talk more about the differences between the two vaccines. I mean, I wasn't involved in any of the technology of either of these vaccines, to be blunt about that. But one of the main differences is really just an approach. It's like the Johnson & Johnson and the AstraZeneca vaccines are using a chimpanzee adenovirus that has encoded in a DNA the spike protein. And so you're actually growing up and culturing a lot of this chimpanzee adenovirus, and an adenovirus is basically a cold virus that you're then growing up, and then you're basically, and they're not replication competent when they go in as a vaccine. So they will go in, and then you'll get a lot of expression from this virus of the spike protein, and so then that kind of allows the immune system to then recognize the spike protein and mount a reaction to it. You're also going to be mounting a reaction to the rest of the chimpanzee kind of proteins associated with with the chimpanzee adenovirus. And so one of the downsides of this is it works well, but next time around it might not work as well because the immune system will recognize and react more to the vector, which is the chimpanzee adenovirus, than it does to, say, the next vaccine that you want to make. Whereas the mRNA vaccine is literally just, as Lynn mentioned, the modified mRNA of the spike protein brought into these vesicles, so it's not a virus, and then the vesicles have been formulated so that they will fuse with cells, and then you'll get expression of the mRNA of the spike protein from the mRNA that's coded there. And so
it's one protein.
Yeah, it's one protein. There's lots of other non-SARS-CoV-2 proteins associated with the J&J and the AstraZeneca vaccine. But you're actually you're culturing a virus in that context that will be replication incompetent, and that's very much different than synthesizing an mRNA and then complexing it with a nanoparticle.
And so if I were a layperson, I'm listening to you thinking I'm a layperson, and if somebody were to say to me, I'm going to inject you with a chimpanzee virus, I would be completely freaked out. But actually, there's no reason to be afraid. It's just a piece of DNA that's going to make proteins that are not harmful to ourselves. But you can see the advantage of the mRNA vaccines because they're simpler in what they're introducing into your cells, our cells. The messenger RNA encodes one protein, the spike protein, and it's a plug-and-play development in the sense that, let's say the spike protein mutates while it's infecting the human population so that the current vaccines are not as efficacious, then we just need to quickly develop another vaccine using the same technology, just a slightly different mRNA sequence, and introduce that into people as a new vaccine. this can also be done for the flu. What's been interesting is because people are masking, there's really been no flu season this year. And so maybe the United States will change their view of using masks more like the Asian populations do. So I have Asians training as postdocs in my lab, and when they feel sick, not sick enough to stay home, but they don't want to run the risk of infecting any of us when they come to work, they'll wear a mask. And if you go to Asia, you see lots of people wearing masks, either because they have the sniffles or they just don't want to get somebody else's sniffles. So who knows what the future will bring?
So there's also with like herd immunity, how do you see that possibly being, do you think like that'll be obtainable with the vaccines that are out currently, or do you think that the virus is going to mutate enough where maybe the current vaccines are not going to be as effective and we might have to continually get new vaccines? So I guess what are your thoughts on herd immunity, if that is an obtainable goal?
That's a Josh question.
I think it's definitely obtainable. The problem is you don't really know what the threshold for human herd immunity is actually going to be. It's like, it's definitely attainable, but it's, whether it's 65% would be optimistic, whether it's 85%, that would be pessimistic. Those are really big ranges. And a lot of this has to do with respect to how long how long are you protective with respect to the vaccine? I mean, you mentioned some of the factors. How long are you protective with respect to if you actually get infected and you're not vaccinated? And then how transmissible is the virus? And so a lot of these are still major questions that we don't really know. There's no question that herd immunity can happen. And a lot of it is just kind of shortening and cutting off the avenues of replication for the virus, because the more people that are immune, the less transmissible it is. The more people that are wearing masks, the less likely people are going to continue to transmit the virus, and it will start to die out. But right now, as far as the variants are outcompeting kind of the wild type virus. But that doesn't really tell you that these variants are way more transmissible from one person to the next. It just means that if you have a mixed population of variants and wild type, the variants are going to be doing slightly better. That doesn't mean that if I sneeze here, or if if I'm talking to someone, that it's going to be like tenfold more infectious. It just means that in the bodies that it's doing better. So it's really hard to project that this is actually killing more people or transmitting faster. That evidence will come, but right now it's not really clear. And so far, they're seeing so frequently, the easiest way to look at immunity with respect to the variants versus the wild type virus is to actually look at if you take immunized people, or if you take people that were exposed to SARS-CoV-2 and take some of their blood, they're going to have antibodies in their blood that are capable of inactivating the virus, rendering the virus not infectious. And so what they do is they actually dilute the blood down, and then say, okay, at what dilution does it actually inactivate the virus? And so, at like a 1 to 500, with the wild type virus, it inactivates the wild type virus. So you're diluting it 500 times. However, maybe it's a 1 to 365 dilution with the variants. And I've seen some of this data. And so yes, in that instance, it's less, the blood is less effective because you have to dilute it. You have to dilute it less in order to get inactivation of the virus, but it's still pretty effective. So it's a little bit of a challenge because, yes, scientifically, if that's statistically robust, it's less effective, but it's still really darn effective. So it's a little bit tough because people hear, scientists say, okay, we're getting a little bit lower neutralizing antibody titer, and everyone's freaking out, when really that neutralizing antibody titer is still really good. And it's still really protecting from like severe disease, which is the most important thing.
Right. So when the mRNA particle is injected into our bodies and makes the spike protein, there's more than one region of the spike protein against which we raise antibodies. So if one of those regions has mutated, we still have antibody against a region that hasn't. And the idea behind the vaccine is to decrease the severity of disease. And it does that despite there being variants.
So could you talk about maybe like any sort of like misconceptions that you might have seen in like the news media with regarding the mRNA vaccines? Is there anything? Yeah.
I think what I hear from people who don't want to take the vaccine, and some of these people are in the medical field, not the clinicians, but at other levels in the medical field, is they feel the vaccine was developed too quickly and they don't trust it. But as we've already talked about, this has been a development that has gone over decades. So I don't have that fear at all. I mean, I've taken the vaccine, Josh has the vaccine, and maybe with the second dose, I certainly didn't feel so hot for 36 hours, but that means my immune system was working and it raised the antibodies I need for protection. And as you probably also heard, the medical history in this country has not been pristine in terms of if a person was going in for clinical treatment or diagnoses, if that person weren't savvy enough or educated enough to ask the appropriate questions, and they might not have gotten answers that they could have used to make an intelligent decision about whether or not they wanted to undergo next steps that were being advised by someone in the medical field. So I think that history has improved a lot, and that's really good, but there's still a fear factor about past events that have taken place that were not according to the medical code of.
There was unethical stuff that went on in the 50s and in the 50s, the 40s, 50s, and 60s, like just terrible stuff.
Terrible.
And there's certain communities that still have a lot of distrust as a result of that. And and so it's hard because ethics has come an amazingly long way from then, but there's still a lot of damage that was done 20, 30, well, more like 30, 40, 50 years ago at this point. But
more than 100 years ago. It's whoever was underprivileged in a community, in a society, let's say you were jailed in the 1800s, you were fair game to basically medical experimentation. And it just goes on.
Okay, great. Thank you. Liz, is there anything else you'd like to ask?
Yeah, I was going to jump in with one question. Just going off of like side effects, could you speak to like why certain people get more severe side effects than other people?
That's a Josh question.
I mean, to a large extent, we don't know necessarily the exact mechanisms for the differences. We just know that these differences exist and that people respond differently to normal viral infection and to vaccines. And so there's a lot of research going into trying to understand the exact mechanisms of this diversity, and they're complex and overlapping. And so it's like there's almost like different immune programs that can be activated under different contexts, and that certain people have genetics that are more likely to activate one immune program as opposed to another immune program. And so, we know some of the factors that will make you more likely to go in this one route versus this other route, and different viruses try to make it so you like go into one route versus another route because some are more or less effective at doing this. And so it's a challenging issue. You know, it's like there's a one of these things is a Th1 type of immunity versus a Th2 type of immunity, and there's various kind of genetic factors that will drive these types of responses. But that question is really, I mean, earlier in the document that you mentioned, you talked about one of the biggest challenges with vaccine development. It's really kind of threading the needle of having people to have a really good immune reaction to the vaccine without having like a really like nasty sickness associated with the vaccine that's going to not make anyone want to get it. And they're really trying to project that based upon how mice react to the vaccine. So that projection from mouse to people is a very difficult projection. And you're really projecting both. You're trying to project protection from a challenge with the virus, but you're also trying to say, okay, we're not making people too sick that they're going to be down and out for 2 or 3 days. And so in both those instances, you're then in your phase 1 clinical trial, you're just trying to say, okay, do we get correlates associated? Do we get antibodies produced that will then be able to inactivate the virus, say in vitro in a petri dish? You know, do we have cells produced that will then go after the virus, and that's all good? And, you know, is everyone feeling okay after getting this virus? You know, maybe a little bit of a sore arm is okay, you know, and if it's a pandemic that's killing, you know, hundreds of thousands of people, maybe getting a little bit of a flu-like symptoms for 24 hours is okay. But that's the type of things that you're trying to balance out.
Right, so they're rare, but I think of 2 things that can happen once you get the vaccine. And one is an allergic response. That's rare. And that's why when you get the flu vaccine, people ask on a little questionnaire, have you ever shown an allergy to eggs? Because those vaccines can sometimes be grown on eggs. And the other is what Josh is talking about, and it's called a cytokine storm. So you want to raise the immune reaction, and then our bodies know how to shut it down. So you get your immune response, you develop your antigens, your antibodies, and then your body shuts down that reaction. But in some people, for some reason, it doesn't get shut down. And as Josh said, clinicians are trying to figure out why that is. Both are rare consequences of getting any of these vaccines. And I think that should be noted. In terms of an allergic response, that's why after you have your vaccine, they want you to hang out for 15 minutes. Because if you're going to have a severe reaction, it's going to happen instantly, and it can be easily treated. Easily, right there, and you're done.
Okay, so with like challenges regarding vaccine development, what do you think are some of the biggest challenges that vaccine developers are like facing, either with like just vaccines in general or with specifically the COVID vaccines?
I mean, a big one is just, I mean, it's really just what the vaccines need to do. They need to be protective without causing overly reactive immunity. And so that is the biggest challenge, you know, just safe and effective. And, you know, and you try that out kind of in animal models and try to make sure that's good. But then moving forward, you know, it's always a different going from, say, does it work in animals to does it work in people? And so like Merck tried, you know, they developed a vaccine that was on the exact same, you know, they were just as quick to get it, and it just wasn't as effective. And it's really hard to project, you know, beforehand. You know, Merck has lots of smart people. It's like, and it's just a complex enough process, and our models aren't very good because we don't have, you know, a human immune system and the diversity of human immune systems that are actually out there, you know, that we can just put in a petri dish or in a mouse. And so, as smart as people are, they can only project things that have kind of worked in the past. And so they try to do that, and then they see how it goes. Sometimes you hit a home run with the mRNA vaccine. Sometimes you strike out, and then Merck is now helping out J&J do their vaccine because J&J's vaccine was so much better. So there's just a lot of uncertainty because, you know, when you're dealing with the complexity of all of the humans' immune systems and it's all very diverse, you don't quite know how that's going to play out because you can't test it in that type of population. So that unknown, I think, is the hardest thing.
Well, so I would take a different answer to that question, and I would make it more upbeat and positive, because when I think of what's the hardest, what's a barrier to vaccines in the future, it's the fear of people to get vaccinated. And I think with the development of the mRNA vaccine, where a lot of the technology has been worked out, and the only variable now will be what is the mRNA sequence you want to encapsulate and put into people so that you can make the appropriate viral protein that will make the person immune to being infected by the virus. You know, that a lot of variables have been eliminated. And so I would like the public to be optimistic and to be very impressed with the technology that we have now in place, and to be more willing to take the vaccine to protect us and themselves.
Yeah, I want to comment on that because I was definitely coming at this from like a technical, you know, okay, the most technical aspect of producing the vaccine. But first off, in that regard, these vaccines came amazingly quickly from kind of start to finish, and it was awesome. It really was a great feat. And if you look at the clinical trials, it's like everyone started pulling together, and the amount of people enrolled, and the data just looks stunning. And it has we talk about flu vaccine, but people are now taking antigens associated with cancer, and it's like it's going to be applicable to a lot of different fields.
Right.
Now, when you talk about barriers right now, it's like, and we're right at that cusp where vaccine availability in our country is enough, and the barrier is going to be to people actually getting vaccinated. It's like,
right.
You know, and that's actually right now going to be the biggest barrier to us getting back to normal and to getting to kind of herd immunity is actually just people getting vaccinated. And it's a challenge because the longer it goes on, the more likely there could be some deleterious variants, or, you know what I mean, that could be worse.
Right.
And I don't want to say, it's like, you don't necessarily know that variants are going to be worse. They could actually become less less pathogenic and just more transmissible. But still, we're without getting the population vaccinated, that's actually like a major barrier that needs to be addressed.
And what Josh said in terms of these vaccines being developed very rapidly, he doesn't mean the technology. He means the versatility, having the technology at changing the mRNA sequence to encode one protein versus another. And it can be applied not only to infectious agents like viruses, but also can be used for cancer therapy. If your cancer cell is making a certain cell surface protein that your normal cells don't, then one can raise an antibody to react with that protein and kill the cells. That's what immunotherapy is all about.
Okay, so I think I have one more question. Do you think there is like one preferred vaccine over the other, like with the ones that are available right now? So I think mostly just Pfizer, Moderna, and J&J. Do you think there's one that's maybe people should get more than the other, or do you think they're all relatively the same?
I think if you have an opportunity to get a vaccine right now, you should take it.
They're all very effective. They're all very, very effective, and so you should just take whichever one is available to you. I mean, we've been talking about the advantages of the mRNA vaccine, but the J&J with a single dose is amazingly effective. And so it's really whichever one is available is the one that you should take. And they all have great safety profiles. They've been, studied extensively, and yeah, so. Elizabeth did just ask a question about about how we would handle talking with someone who is reluctant to get the vaccine. And I started I started to say that first I would listen to their concerns, and depending on those concerns, but from what I've heard, are concerns about, how quickly it may have been. And so one of the major things that slows down, vaccine production, or say test vaccine, starting from start to finish, is kind of the ability to accrue patients into a study and see whether they are protected over time or not. So for example, if you have, let's say you want to test a vaccine to herpes simplex virus, you need to, let's say, we come up with a vaccine. And so what you need to do is you need to enroll people that are willing to get vaccinated, okay? And then you need to follow them for a length of time to see if they're going to turn positive for herpes simplex virus. And you got to see if they're going to become, if they're going to have any side effects. It's not like you're going to be challenging them by giving them herpes simplex virus. You just need to see if they're going to actually acquire it on their own. And so such types of vaccine studies take a really long time because you have to follow the natural, you have to have enough people naturally get HSV1 positivity over a time period of, say, for example, years, in order to say that your vaccine is protecting your population that actually got it. Now with coronavirus, because there were so many people that hadn't had it, and because the exposure is so kind of rampant, that you could actually do that type of trial for efficacy much, much quicker. Okay, and so whereas for lots of the other things that we do trials on, that is not the case. Even for HIV, it's not like you're going to give people HIV. So what you need to do is you need to vaccinate a group of people and then not vaccinate another group of people as your controls. And then as enough of your controls just happen to get exposed to HIV, then you can actually start to see whether you're being protected. So that's a major challenge in dealing with the clinical efficacy of vaccines. And so, but with coronavirus, because it was so widespread and we had such a population of people that hadn't seen it, we can do that much faster. But yet the actual safety profile, that's a lot easier to do quicker, because you can see relatively quickly if you're going to have like a negative side effect. And so that part hasn't really been compressed. It's just the part of, it's basically the accessibility of people going from not having SARS-CoV-2 to having it, and seeing how that can be protective. That went much faster. Does that make sense? And so that's one of the things that I try to explain to people, like, hey, look, the efficacy, the safety profiles is about the same as how it's done. It's just like now we can do the efficacy much faster because we have large populations of people and large populations of people that want to be vaccinated. If you're like, hey, do you want to do like you want to get a HSV vaccine? Some people are like, I don't, you know what I mean? Like, no, now if you're going to say, well, I will give you SARS-CoV-2 vaccine, there's tons of people that wanted to sign up for those trials. So it just means that they can go much, much faster.
Another example of a vaccine that you've probably gotten is papilloma, right? So many young people have been vaccinated for papilloma. There are, I think it's 3 strains that cause cervical cancer, and this is a really good thing. You've been vaccinated, right?
Lynn, you're cutting up. I don't know if you're connected, but you might want to plug your computer back in if
You hear me now?
it's not good. So
Yeah, I have. All
you may be
right.
back now.
Just be positive.
Okay, we lost her. The other thing I feel like that we try, that I try to do is just talk about how everyone who works on the vaccine and the doctors, it's like we're all getting, it's like we all believe very strongly that this is a good thing to do. And so, and it's also the way that we're gonna get out of this kind of mess, and vaccines have been given for years, and it's like it's like all the technologies, the vaccine technologies are not new. They're tried and true, and any semblance of any like kind of looking at associations with negative effects have always, they've never held up to scrutiny. And so the fact that this is a new one, all of the ways that we've kind of come across have been tried before. And so it's like you always need to pay attention, but it's not like it's like this totally new Frankenstein monster. So, but it's hard, right? Because there's a lot of misinformation. There's a lot of independent echo chambers that just kind of reinforce kind of these narratives that are destructive with respect to what the science actually suggests. And whether that's Facebook or so, it's a challenge.
Yeah, definitely, for sure. I feel like definitely with like the previous administration, like politics and science, there's been a lot of like, I guess, mistrust in scientists. Have you noticed that at all with like your research?
I don't think this. It's like most scientists don't interact a ton with the general public in a science way. It's like I when I go to my daughter's soccer game, like it's not like there's a lot of non-scientists there and we're not talking science. But when I'm talking with scientists, with science is typically with other scientists. But there's no question that where science engages public policy, that this is a major issue, and politicians can really shape the nature of interaction of scientists and public policy and science and public policy, and they can do a lot of damage. And I would argue that a lot of damage has been done. Now, it's like, and in some ways, it's like science doesn't have all the answers, right? It's like, and so, and sometimes when we point that out, it's like when we point out the complexities, then that opens other people to be dogmatic about things, even if those are false narratives. And so it's a real challenge. So yeah, it's tough, but I mean, personally, I think Fauci has done a pretty good job just trying to be honest with what was going on, and highlighting what we know and the limits of what we know at the same time, and take that with as you will. But it's clearly a challenge because the public has become a lot more skeptical.
Yeah, definitely. I don't think she's gonna come back, so we might have lost her permanently. Is there anything else that you'd like to add in general? Anything that we didn't cover?
No, I mean, you mentioned in your question what causes these new variants to emerge. And basically, there's just a selective pressure for the things that replicate best, and that could be at the RNA level, that could be escaping, there's a lot of different areas where, certain mRNAs could replicate better than other mRNAs, and certain viruses might have a slight selective advantage. So that doesn't necessarily mean they're more deadly or that they're more likely to be infectious from one person to another, but yet there's always going to be kind of like that selective pressure. If you can get more copies of yourself packaged into a virion, they're going to win out. And right now there's variants that are clearly winning out, but the actual reason why they're winning out is not totally clear at this point. But that is a very common and well understood process that was gonna happen. How that plays out, it's like, I think one thing that's important to realize is it's not the virus's goal to make us sick. The virus's goal or evolutionary's goal is just to reproduce and survive and persist in the population. In some ways, if it didn't make us sick and just reproduced and survived in a population, that would be the best thing for the virus. And so, colds do that really well. And other coronaviruses that are just colds have evolved to do that really, really well, despite the fact that they don't really make us that sick. So it's possible that SARS-CoV-2 could be heading in that direction. We just don't know. And so it's like when people think about evolution, they think about frequently, or at least the public might think about, it's going to evolve into a killer virus. But frequently viruses aren't trying to evolve into a killer virus. They're just trying to evolve into a virus that spreads a lot and that maybe doesn't cause any illness because the immune system doesn't see it as too much of a threat because it's not a threat. So, yeah.